Hello students, my name is Shankar Banerjee. I am the founder and educator at Bioscience Institute. In today's topic, we will be discussing about transport of secretory proteins to lysosome. Now, we all know that most of the proteins, after being synthesized by means of translation within the cytosol, what it does, it enters the endoplasmic reticulum lumen. Within the endoplasmic reticulum lumen, what happens? That particular protein will undergo certain kind of modifications, and among all the modifications, we know glycosylation is the main. During glycosylation, what happens? Certain moieties bind with certain moieties. The glycosylation can be of two types, N-linked and O-linked. I am not getting into the depth of it. Actually, what happens? Two N-acetyl GlcNAc units bind, N-acetyl glucosamine units, 9 mannose unit and 3 glucose unit. It binds with the entire protein. And that protein, after this kind of glycosylation, it is being transmitted from endoplasmic reticulum to cis Golgi. From endoplasmic reticulum to cis Golgi via COP2. This is basic overview. We all are aware of it. Now, when it comes to lysosome or proteins that are supposed to be transmitted to the lysosomes, what happens is this cis Golgi. This is very interesting. This cis Golgi, it bears a special type of enzyme. This cis Golgi, it bears a special type of enzyme which is N-acetyl glucosamine phosphotransferase. And this particular enzyme, it bears a catalytic unit. Within the catalytic unit, this particular enzyme, it captures the proteins that are associated with lysosomal transport. So this N-acetyl glucosamine phosphotransferase will capture the proteins that are supposed to be transported to the lysosome within it. And what it does, it transfers its phosphate to the mannose residue. It transfers its phosphate group to the mannose residues. Where mannose residue? Within the protein. So let me repeat. First of all, the basic concept: after translation, these proteins are entering the endoplasmic reticulum lumen. Within lumen, it undergoes glycosylation. In glycosylation, there lies certain units that binds with it, moieties that binds with it. Among them lies 2 GlcNAc, 9 mannose and 3 glucose. And these, after glycosylation, the proteins are transmitted to the cis Golgi via COP2. Now what happens is, within cis Golgi lies a special kind of enzyme which is N-acetyl glucosamine phosphotransferase. Within that enzyme, they possess a catalytic unit that attaches the specific protein that are supposed to be transmitted to the lysosome. And what it does, it transfers its phosphate group. To those proteins. And the proteins normally receive the phosphate group upon mannose residues. That protein receives the phosphate group upon the mannose residues at position 6. Now what happens? As now the protein, this protein which has mannose 6-phosphate with it, after the formation, this protein, it detaches from the enzyme N-acetyl glucosamine phosphotransferase. So this protein is now separate, is now free. With this protein binds a specific kind of receptor. With this protein binds a specific kind of receptor, which is the mannose 6-phosphate receptor. This mannose 6-phosphate receptor, which is present at Golgi, this enzyme was present at Golgi, that is converting the protein into phosphorylation or into its phosphorylated form, and that phosphate is added to the mannose vesicle. What is the purpose? So that this protein that is supposed to be transmitted to the lysosome can bind with the mannose 6-phosphate receptor, and this mannose 6-phosphate receptor is localized to Golgi. So our protein will bind to the receptor, and after the binding via clathrin AP1 cargo receptor or cargo protein, it will be transmitted via clathrin AP1, it will be transmitted to endosome. It will be transmitted to endosome. So first we have our protein. That protein undergoes phosphorylation by the activity of enzyme N-acetylglucosamine phosphotransferase, and this protein, which is now phosphorylated at its mannose residue, can bind with its receptor. That receptor is present none other than in Golgi, Golgi, not Golgi, and this entire thing is transmitted via clathrin AP1 to the endosome. Now let's see the picture. We have the entire idea. Let's see the picture. So this is our Golgi network. Within that Golgi network lies our mannose 6-phosphate receptor. Now our protein that is already phosphorylated at mannose residue, our mannose 6-phosphate receptor binds with it. See, this kind of binding takes place. And after binding, what happens? Via clathrin, it is transmitted finally to late endosome. Now we all know late endosome has a low pH, which is near about 5.5, and the binding of mannose 6-phosphate receptor with the protein takes place at pH 6.5. So pay a close attention. This binding occurs at pH 6.5, but late endosome, its location has a pH of nearly 5.5. So this low pH, the mannose 6-phosphate receptor cannot bind. The mannose 6-phosphate receptor cannot bind or remain itself bound to the protein. And as a result, the protein will be detached. And as the protein is detaching, this mannose 6-phosphate receptor will again be recycled from here to here. So this is the overall mechanism. First, we have got our protein. That protein enters endoplasmic reticulum. It undergoes glycosylation. After glycosylation, transmitted to Golgi via COP2. Within Golgi lies a specific type of enzyme N-acetylglucosamine phosphotransferase. Its catalytic unit, it captures the protein, and that protein gets phosphorylated at its specific mannose residue. After getting phosphorylated, this protein with mannose 6-phosphate in it binds with the mannose 6-phosphate receptor, and this mannose 6-phosphate receptor is present none other than within the trans Golgi. After this kind of binding, the protein and the receptor, it forms a close association, and then they are transmitted finally to the late endosome. Now this binding, it takes place at pH 6.5. But its final location, the late endosome, which will eventually form the lysosome, it has a pH of nearly 5.5. So this low pH cannot hold our protein with that of its receptor. Hence, they will be detached. And the mannose 6-phosphate will again be recycled to initiate the process once again. So this is the overall process of lysosomal transport of secretory proteins. This is how the mechanism works. And the clinical condition that is associated with it is eye cell disease. Very, very important, very question prone area when it comes to CSIR-NET. A severe lysosomal storage disease where enzyme N-acetylglucosamine phosphotransferase is missing. The actual enzyme that is creating this entire process, the N-acetylglucosamine phosphotransferase within this Golgi, is missing. And as a result, what will happen? Hence, no mannose 6-phosphate residue formation. And as a result, there will be no transport of secretory proteins into lysosome. So this phenomenon is called eye cell disease, where the enzyme N-acetylglucosamine phosphotransferase is completely inactive. As a result, mannose within that glycosylated protein will not be phosphorylated. Hence, mannose 6-phosphate receptor cannot bind with that protein and cannot be transmitted. So this is the overall procedure, and this is the clinical condition associated with lysosomal transport. If you find this video interesting as well as informative, do share with all of your batchmates. And always remember, stay focused, build concepts and track progress. Me, Shankar Banerjee, founder and educator at Vital Science Institute, signing off.